In a groundbreaking discovery, researchers from the University of Houston College of Pharmacy have identified key mechanisms behind skeletal muscle regeneration and growth following resistance exercise. This finding paves the way for developing targeted therapies for various muscle disorders, such as muscular dystrophy, which affect millions of people worldwide. Skeletal muscles are essential for numerous bodily functions. They control breathing, regulate whole-body metabolism, and enable every movement we make, from walking and running to sitting, standing, smiling, and even blinking. Understanding how these muscles regenerate and grow is crucial for improving overall health and treating muscle-related disorders. Skeletal muscles are formed during embryonic development by the fusion of specialized cells called myoblasts. In adults, muscle regeneration is driven by muscle stem cells known as satellite cells. After an injury, satellite cells proliferate and differentiate into myoblasts, which then fuse with each other and injured muscle fibers to repair the damage. However, in many muscle disorders, this regenerative capacity is diminished, leading to muscle mass and function loss. The University of Houston researchers discovered that the inositol-requiring enzyme 1, or IRE1, is crucial for myoblast fusion during muscle formation and growth. During muscle regeneration, IRE1 augments the activity of Xbox-binding protein 1, which in turn stimulates the gene expression of multiple transmembrane proteins required for myoblast fusion, said Ashok Kumar, Elsay and Philip Hargrove, endowed professor of pharmacy. Increasing the levels of IRE1 or XBP1 in muscle stem cells outside the body, followed by their injection into patients' muscle tissues, can improve muscle repair and reduce disease severity. We also found that augmenting the levels of IRE1A or XBP1 in myoblasts leads to the formation of myotubes having an increased diameter, added Kumar. This discovery has significant implications for treating muscle disorders. Size is very important for muscle. Muscle grows only in size, not in number, said Aniket Joshi, a graduate student in Kumar's lab. Larger muscle cells generally work better, lifting more weight, running and walking faster, and improving overall metabolism. This can help prevent various diseases, such as type 2 diabetes. By enhancing muscle regeneration, this research offers hope for those suffering from conditions like muscular dystrophy. This new research builds on previous work by Kumar's team. In 2021, they published a study in the eLife journal describing the role of the IRE1A XBP1 signaling axis in regenerating healthy skeletal muscle after acute injury and in models of Duchenne muscular dystrophy. They found that the IRE1A XBP1 signaling axis plays an important cell-autonomous role in satellite cells. Along with Kumar and Joshi, postdoctoral fellow Meiricris Tomaz da Silva and research assistant professor Anirban Roy conducted the research. Other contributors from the University of Houston include Micah Castillo, Preethi Gunaratne, Mingfu Wu, Yu Liu, and former postdoctoral fellow Tatiana E Koike, along with Takao Iwawaki of Kanazawa Medical University, Japan. This ongoing research continues to explore new ways to enhance muscle regeneration and treat muscle disorders. The identification of key mechanisms in skeletal muscle regeneration marks a significant step forward in muscle disorder treatment. By understanding and manipulating these processes, researchers can develop targeted therapies that improve muscle repair and function, offering hope to millions affected by muscle-related conditions.